Hi everybody. Now we're going to be talking about translation. So we did transcription, that happens within the nucleus. Translation, of course, happens at the ribosomes in the cytoplasm. So it's the second step of gene expression. There are three stages as well: initiation, elongation, and termination. It uses information from the mRNA molecule to direct the different types of amino acids. So how many different amino acids are there? Well, you might remember that there are 20 different types of amino acids in humans, and they're being directed by the tRNA. So translation begins when the rRNA, that's the ribosome, moves along the messenger RNA molecule in increments of 3. So the messenger RNA has joined on to the rRNA. It looks at the messenger RNA in 3 nucleotides, and that's called a codon. So adding an amino acid every time it sees a different codon, the step of 3, until it reaches a stop signal. A newly formed polypeptide is then released. So we're going to be talking about this in much, much greater detail. First, we're going to be talking about something called the genetic code. So genetic code, I just told you that the mRNA rests on the ribosome so that 3 nucleotides are exposed. And those 3 nucleotides is called a codon. So when you do excuse me. When you figure out how many possible combinations there are, you'll notice that there are 64 different possible combinations. So AAA, AAT, in this case we're going to be looking at mRNA, so it's going to be AAU, AAC, AAG, and then ACA, AGA, AUA, and so on. So there's 64 different possible combinations. Those combinations are called a triplet. There are 20 different types of amino acids, 64 combinations, so that means that some of them are going to be replicated. And this 64 combinations, that's called the genetic code. So as each bit of mRNA is exposed, a molecule of tRNA with the complementary 3 nucleotide sequence called the anticodon binds to it. This tRNA carries a specific amino acid which is added on to the growing polypeptide chain. The ribosome moves to the next 3 nucleotides, codons, and a new tRNA with a corresponding anticodon sequence attaches and adds another amino acid onto the growing polypeptide chain. We're going to go into that process in more detail. Right now we're going to be talking about the genetic code. So let's talk about genetic code. So it's the same for all organisms, very few exceptions, just in some archaeas. Every time you get UCU, it codes for serine, that's the amino acid. UAC codes for tyrosine. What does GCU, AAG, AGA code for? Well, we use the genetic code. In your textbook, you can find this genetic code right here. Let me focus. There we go. Genetic code. You do not need to memorize it, but you do need to know how to use it. So further along in your note right here, you can see a nice simplified version of it. We've got one side, we've got a top side, and then we have a third side. And what you do is you read the first letter, second letter, third letter. So let's take a look at those mystery ones. GCU. G, C, U. So G, C, U. Alanine. ALA stands for alanine. AAG, AAG. There we go. Lysine. And finally, AGA, AGA stands for arginine. There are nonsense codons. So there are codons that cause termination. And whenever UAA, UAG, or UGA get read, it means that's the end of the gene. And then there's also a start codon, and that's AUG. So the first time AUG is read, that's where the ribosome will actually join on. So AUG, every time afterwards AUG is read, it stands for MET, methionine. You don't need to memorize the genetic code, like I said, but you do need to know how to use it. Thanks.